a research strategy where computer and related applications can be taken into consideration. So, as we are somewhere revolving around the concept of the computer and computer applications, so how these computer applications can be considered whenever we are deciding any research strategy, whenever we are discussing any topic, whenever we are formulating, suppose, any proposal for any kind of the research. So, where we can have a space, there we can consider these all sort of the computer applications. Moreover, we are also going to have discussion upon the different resources which are available online, offline, okay, open sources, resources, or say commercial sources, which are uh, available on net as well as off net. So all these online resources, whatever, as I said earlier, these could be the commercial one or say open sources, mm, are widely available. So few of the slides are there in forthcoming discussion, so which will give you an idea how these sources can be directly downloaded from the web sources, how to exploit all these resources, and also we are going to discuss few of the example lively, and how these tools can be used or exploited. We are going to see a live demonstration for these tools. Okay, so let us begin. So what do you think? What could be the fundamental role of a responsible pharmacist? Okay, so it is a fundamental responsibility. Rather, we can say that to develop a safe, effective, and stable drug dose formulation. Now, in order to develop this safe, stable, or effective dosage form, what usually you do in your laboratory or during your course or during your program? So you usually try to practice different formulations which have been given in your lab manuals. Also, you are trying to perform your project, which could be a task for your final year semester, and like that you try to these could be the possible areas where you can practicize, where you can try to develop this safe, effective, or stable dosage form. Actually, it is a task which is usually concluded in the pharmaceutical industry, but as we are going to discuss these particular pharmaceutical applications, or say these online applications for pharmaceutical purposes, we are going to confine our discussions around the project work, because this is a very big task. Most of the time, most of the students, they usually face that the project work is always a huge task, and something new has to be done during the project work. Likewise, they are having their own framework and thought framework, and because of that, most of the time we usually see that most of the students are very anxious, very panic during the performance of their own project. Okay, no issue for your say the practical formulations or whatever are there. These are various formulae have been given in your lab manuals, and simply we are looking at that formula. We are, whatever be the procedural steps have been given in the manuals, we are simply following those all steps, and we are developing that particular formulation. So these are the practices at the UG level or PG level we are doing to develop or to become a responsible pharmacist, or responsible professional pharmacist rather than major issue. What we are going to look at about the project work, how to devise a strategy, as I said in our overview, how to devise a strategy, how to perform this particular project work, and what are the different spaces available where we can extend our work, and how we can complete our project, and that could be even the part of the present discussion. Okay, so let us discuss how to formulate a safe, effective, and stable dosage form, and fulfill the role of the responsible pharmacist. Now, in order to start with the actual discussion, let me get the answer for how this particular drug dose formulation can be formulated. So this is a very general approach, and this is what we can say, it is a pre-formulation protocol rather, and how this particular pre-formulation protocol can give us an idea how these excipients can be selected, what could be the possible proportion of each excipient, how different packaging materials could be selected, and not even that, whatever be the claims we are making upon the label of that drug dose formulation, all these claims, how to formulate even all those claims, how to write the instruction, how to write the how to write the warning uh, upon the label of any particular drug dosage formulation. Okay, so this will come from this particular slide. All the answers for this may come from this particular slide. Okay, so let us discuss. So most of the time we do have our own API, so that is the active pharmaceutical ingredient, and this particular API may be synthetic or say semi-synthetic, or most of the time it could be obtained from the natural resources. Okay, so this API is usually along with us, and we try to develop. We used to discuss with your guide that I want to develop this formulation, I want to develop that formulation, or even we are uh, going through the development of the few of the cosmeceuticals. Okay, so this is as per the API, and based upon that API we used to develop the formulation. But what is the usual approach, how to go for the actual drug dose formulation? We need to determine few of the say the characteristics of that particular drug, so that effective drug dose formulation can be formulated. So first of all, we are working in a four major areas which are revolving around this particular API, and that could be the part of the pre-formulation protocol. So first you tend to go for the determination of the organoleptic property. So you are well aware, not even there is a need to discuss what kind of the organoleptic properties are there: color, odor, taste. Okay, suppose as far as concerning the taste, if your active constituent is very bitter in taste, what you usually do, you try to go for the selection of any sweetening agent, just to mask the bitter taste of that active compound. So this is the first say what we can, the selection of the inactive constituent, that is your excipient you have selected over here. Say for example the sweetening agent. If for example, suppose in your study you come to know that the sweetening agent what we have selected is not having the good compatibility with your active constituent, then you can mask the test of that particular API simply by using the various polymers. Okay, so various coating techniques can be, particulate coating techniques even can be used. So there, if the sweetening agent is not working, then you can go for the selection of the polymer. Okay, so not only that, we need to determine again further its composition too. That means, see how we have selected, we are having one API. Now we have selected either the sweetening agent or any of the particulate polymer for the coating, and this, say for example, polymer or sweetening agent could be first of all our inactive constituents. Okay, even we can decide its proportion. This was all about say organoleptic property. Then we move towards next segment, that is the bulk characterization. I'm not going to discuss in detail the complete topic of this pre-formulation, just for the sake of simplicity and just for the sake of the concept of the drug dose formulation. I'm giving an overview upon this particular pre-formulation study. Then next, here we are having say the selection of the inactive constituents, say the sweetening agent and your polymer. Now here, in case of the bulk characterization, we usually go for the determination of say your hygroscopicity. We tend to determine the phenomenon of the polymorphism. You usually go for the determination all the flow characteristics. Okay, so we are quite familiar with the flow characteristics. Okay, so angle of repose, bulk density, tap density, Hausner's ratio. We're quite familiar with that. So whenever we are determining, say, these flow properties for this API, at that time also we are having a tremendous opportunity to get selected few of the inactive constituents or the excipients. Suppose the API what you have selected is not having a good flowability during the determination of the angle of repose, you are going for the glidant so that the flowability could be increased. So glidant is what again say your one more inactive constituent, and it's further again its proportion in what concentration it has to be used during the preformulation. That could be the determination of the proportion, and likewise you can have one more opportunity to select one more inactive component of your drug dose formulation. Suppose your API is suppose granulated with say wet granulation method or dry granulation method, then again the granulating agent is coming ahead. Okay, so we need to select that particular granulating agent. Okay, we must select even the adhesive for that. So likewise, one by one we are selecting few of the excipients likewise. So suppose when we are going for the solubility determination, as all of you are well aware that solubility is very important phenomenon, because instead of having a good solubility, it's very difficult to get the optimum bioavailability of the particular drug. So in order to have a better bioavailability, there must be a good sort of the solubility. So many enhancement, solubility enhancement techniques we have come across, we have discussed several times in different subjects. So here solubility is very important to determine, and if there is a poor solubility, then you need to increase its solubility. What kind of the insights can be built whenever you are determining the solubility? See, there are several parameters what can be determined in context with the drug dose formulation. The first, if you are determined the solubility, and if you have found the solubility of the particular drug is very high at say alkaline pH or say the acidic pH, then you can target the compartment of the drug. Suppose, for example, how these entry-coated drug dose formulations have been formulated, because that particular drug has got a good or better solubility in at alkaline pH. That's what we have prepared it in an enteric drug dose formulation. Now, this is what does say the impact, or this is what the significance of this particular determination of the solubility. We are not only getting the compartments, we are not getting the idea about the selection of the compartment, but most of the time we are getting so many say the selection of even the what type of the drug dose formulation can be built up. Suppose you have done all the efforts to enhance the solubility, and at the end you found that the solubility is not able to increase at all. Then one more way is there to develop a suspension drug dose formulation. Okay, so this is what that means. The solubility is not giving you only idea about the selection of the compartment of the body, but sometimes these the studies can give us an insight how to select what type of the drug dose formulation can be built up. Okay, so if the solubility is not increasing at all, no issue, we can go for the suspension drug dose formulation. Likewise, every area is giving us something idea about the drug dose formulation. Now, say for sake of the stability, why to determine the stability? Just we need to claim the shelf life. We need to claim the expiry date upon the label of particular drug dose formulation. So, how to claim? So we need to do its stability study. Okay. Now, there is a big magic while determining the stability of a particular compound. Just wait for a minute. Is my screen visible? Am I audible?
Yes, sir.
Is my screen visible?
Yes, sir.
There is a big magic hidden while performing the studies upon the stability of a particular drug dose formulation, or say even upon the active pharmaceutical ingredient. That is the stability study of the API. You can keep several samples for the stability in different containers at different temperatures, at different humidity levels, and see how you are getting an idea about the selection of the container. Suppose you have kept the sample at in a glass container, in an aluminum foil, or say in any one of the plastic container, and you come to know that whatever be the sample what we have kept in a glass container, that too in a amber color container glass, that sample has got a maximum stability than that of the other sample. So what you can interpret from this particular stability study, you can interpret over here that the sample which what you have kept inside the amber color container has got a maximum stability. That means your sample can be other samples what you have kept in other materials, and these samples are not have a good stability. And here you can select the type of the container. That means you can what you are writing the upon the label claim that store in a well closed amber color container. Okay, tightly closed amber color container. So why do we write this particular instruction? Because during the stability study we have studied that this particular API has got a maximum stability in a amber color container. Another interpretation can be built up over here that your active constituent may be photo sensitive. If you are keeping it say in a transparent glass container, it doesn't have that much of the stability, and the reason is that it could be photosensitized and it could get degraded over a period of the time. So this is what the stability can give us insight. Stability study can give us an idea in selection of your drug container. Now in the same study, the same sample can be kept at varying temperature conditions as well as the humidity conditions. And here again, you can come to know that whatever be the sample, what you kept at, say, at a cooling temperature, say 2 to 8 degrees Celsius, that particular sample has got a maximum stability rather than the sample what you kept at a room temperature or any other temperature. Now, here you can again make a label claim that store the drug formulation, particular drug formulation at a cool temperature. Because in stability study, you have come to know that the this API has got a maximum stability at a cooling temperature. So, likewise, you are getting enough data, and this during the pre-formulation study, and this enough data can be used successfully for preparing a particular drug dose formulation. So this is a brief overview how a particular drug dose formulation can be prepared, how to select the excipient, how to select its proportion, okay, how to select the type of the drug dose formulation to be developed, whether it is a solution, whether it is a tablet, whether it is a suspension. Okay, so everything, every idea can be withdrawn during the study of this particular pre-formulation protocol for a particular active pharmaceutical ingredient. Okay, now uh, where do you have whatever we have right now discussed, so these all discussions were revolving around the basic research what we are doing in our laboratory. Okay. So being student at the UG level or PG level, we can extend maximum up to the preclinical. Okay. So this is not an our area right now, because this is all work assigned to various pharmaceutical industry setups, various setups of the R&Ds of different pharmaceutical companies, even the CROs. So this could be the possible role of the CROs. And when you in your professional life, when you will start your professional life, at that time you could have excellence in each of the particular segment. So we are not going to have deliberations upon these particular phases, but this is the entire what we can say the drug development or drug development process. And in this drug development, we are usually moving, we are usually revolving around only the basic and early research. At our PG level, maximum we can extend our work up to the pre-clinical level. But how to do a basic research, or say the early research, or even say the preclinical research? How to devise a strategy so that we can have a better proposal for a particular research, and how we can have some sort of the outcomes which are evidence based, having a full proof support of the evidence, so that whatever be the hypothesis we are building, that hypothesis has got good support. And we can say that whatever be the work what we have done in our research, uh, is it having any sort of the value or not? So this is a common strategy if you want to focus upon. Okay, so in this particular slide, we as we have discussed that the basic research can be done maximum up to the preclinical, and how a strategy can be devised so that we can have a better research proposal. And mm, these whatever encoded on this particular slide, this is a very common phenomenon, this is very common strategy. And how this common strategy can be used to have a complete exploitation of the different resources, that we are going to see in a forthcoming slide. So first of all, we used to select the disease upon which we want to develop a suitable drug dose formulation. Okay, so why that disease has occurred, what went wrong because of which few of the receptors are not performing well, and because of that the consequence of that non-functionality of the receptors has raised that particular disease. Okay, so we need to identify the different receptors which have been involved in the particular disease. Once we identify the receptors, there is now turn to identify the standard drugs available for the same. Say, for the sake of the example, if you are suffering from any hypertensive disorders, then suppose, for example, atenolol could be the standard drug. So we need to first of all select our standard drug, which is established into the market for the given disease. Then we can select a similar molecule. The process of the selection of the similar molecule could be your research. We don't want to do anything. Diseases are the fixed, receptors involved are the fixed, all the standard drugs, these are approved drugs, so we don't have any scope to do anything in this particular case. But being a UG or PG student, here we can have enough scope, so that we can better work upon the similar molecule. We can develop, with the help of the knowledge of the chemistry, we can develop a molecule which is not exactly same but similar to that of the standard drug what we have considered for the particular disease, so that we can at least do. So the last step could be the selection of the similar molecule. In one slide, all these words seems to be very scanty in a very very small say the size, but the task involved in each of the category is huge. Okay, and that is what the basic research to develop a new molecule is very tantalizing task. And let us discuss how this particular segment can be selected and how can be a better strategy for research can be built up. Okay, so first of all, why do we lose the ease in any kind of the disease? Okay, so this that is what the loss of or the way we are losing the ease, which can leads to the disease. Okay, so it's only because of the alteration or the change in the function of the specific enzyme or the receptor, uh, which is involved in that particular disease. And what is the reason? It could be because of any genetic abnormalities or few of the physiological or say the environmental stressors are involved, which may cause this particular alteration. Okay, so and ultimately few of the symptoms are arising, and these symptoms are nothing but the clinical manifestations of that particular disease. Okay, so this is what the disease could be, and it is only because of the receptor rather. Okay, now you are having the drug target and the drug. So right now we have discussed what is the disease, the functional, say the key role in any disease is usually played by the receptor involves, and these receptors could be your drug target. Okay, so in alternatively we can say it as a lock. Receptor is a lock. Okay, so this particular receptor usually used to bind with other molecules to perform numerous functions required for the maintenance of the life. Okay, so when this particular function or when the performance of this receptor is not going well, then the quality of the maintenance of the life is not up to the mark, and you may suffer the disease, and that is only because of the drug target. Okay, so many different and very what you complicated terminologies are there here, that is the drug identification, target identification, but we are not going to discuss those all things. So we are going to we are trying to stick up only to the basic discussions which is revolving around the our pharmaceutical drug research. So this is what about the drug targets. So drug targets are your receptors. Alternatively, we are calling it as a log, and the drug is what your small molecule. Okay, so which could be the possible key? Okay, designed to bind, interact, and or modulate the activity of the specific receptor and enzyme. And this modulation can functionalize or can restore the function of the receptor, and by this way we can get rid of the disease what we were suffering. So this is what the two terms which are very important to know in the process of the building of any research proposal. Okay. Now, what we can do for the drug target, see these are a very fixed number studied receptors are there. There is a online repository is there, and in that repository you can see all the information regarding all the receptors. Okay, so we can say that the number of the drug targets which have been studied, which are there available online in the form of the open source or say the commercial source. Okay, Mm, so these are the fixed one. But as far as concerning the drugs, you can get an idea about this particular suppose the selection of the drug from this particular slide. Okay, so where is your tremendous opportunity to work upon? So in the process of the drug discovery, there is one term that is chemical species. And what do you mean by the chemical species? These are the total number of the chemicals in the universe. Okay, so these are total number of the chemicals in the universe, which include the natural and lab made organic molecules. So what is the particular number of the chemical which is available in a universe? You cannot even the count that number 10 raised to 60. So it's very difficult to figure out this particular mathematical expression of this particular chemical species. Okay, so out of these all chemical species, only 1700 are being clinically tested and FDA approved. Okay, and there is your opportunity starts now, because as per the general database of the 2070, approximately 166.4 billion molecules up to 17 atoms predicted to have the stability and accessibility. So this is a molecule, and the bunch of 17 atoms may be involved in the single molecule. Likewise, 166.4 billion molecules are there, which can be assessed for its stability and accessibility. So very huge number again. So one more data, online data is available, that is the PubChem, and upon that, approximately 70 million drugs or 70 million chemicals or the compounds have been registered, and you can have opportunity over here to access the PubChem data for getting information of these 70 million compounds. Okay, so these compounds are the chemicals and have are deserve to have the drug like efficacy. Not essentially that all these chemical 70 million registered compounds must have some sort of the therapeutic efficacy. It's not like that, but at least we the being a chemical compound, it may have capacity to act as a drug compound. And being pharmacist, while developing a drug dose formulation, we can get a compound which is having a structural similarity. Now we are there at the point what we have discussed in a previous slide, that is what identify the standard drugs available in this for the same disease and select the similar molecule. So there is the data, online data available with us. For example, is the PubChem, and from that PubChem, we can get a similar molecule. This is a registered compound, and we can have opportunity to make the changes upon that registered compound, and we can make it similar to the standard drug available for the particular disease. So this is our first approach, what we can do. Now being student, we can what you can do as a part of the project, you can decide a strategy to search upon your what you can say the PubChem. So how to utilize the tools available upon the PubChem. If you look at the website for the PubChem, and what are the different keys which are there, or what are the different buttons which are available upon the PubChem web page. So those all buttons give some sort of the information about the drug what we have selected, about the compound what we have selected, and what we want to make upon the change, how we are going to change that compound, how we are going to apply the knowledge of the chemistry, how we are going to apply the knowledge of the structural activity relation, so that that particular compound can have a similarity, at least structural similarity, with the standard drug for the disease what we have targeted of. Okay, this is what now being student, what I said, being student, you can develop a strategy to study the PubChem web page. Okay, so you can build up your project in such a way that if you're a UG student, you can have the title, right, and that the different tools and keys available upon the PubChem. So each key can give us a very huge informational data regarding the each compound. Okay, so all physiological properties, all physicochemical property, even the ADME profile could be available. And as I said earlier, these are the simple compounds, not necessary that the ADME profile must be checked. So simply whatever be the ADME profile of that particular compound is available, that can be predicted one by using again several online tools. Okay, so like that, a different what we can say the project strategies can be built in context with such kind of the online tools or online repositories which are available for the different compounds. Okay, now how many unexplored chemical spaces are still there? Okay, so approximately 10 raised to 20 or 10 raised to 24. Now here, very important, drug like molecules are yet to be explored. Now here is what your opportunity lies. You can select this compound, and what is the number? Again, a huge number is there, and these compounds can be selected because these are a drug like molecules. These are what? These are the drug like molecules and tend to have or deserve to have capacity to show some sort of the therapeutic significance, provided few structural or conformational changes must be made upon these drug like molecules, and that is what our role is. Now, if I'm talking upon this particular very basic research, then you may feel that this is very difficult task, but this is not at all a difficult task. One of the example of the established, say the drug molecule is the atorvastatin, and that particular atorvastatin, whatever the billions of the market is there for the atorvastatin, that molecule has developed using this in silico different tools. Okay, it was predicted that this atorvastatin could have this particular drug efficacy in the cardiovascular diseases. So simply it was the predicted, and that prediction now came into existence with several allied researchers. But it was the computational tool. It These were the in silico tools which were used to predict the efficacy of the atorvastatin. So without doing any experiment in the laboratory, simply the activity of the atorvastatin was predicted at that time with the help of the in silico tool. And later on, few of the experiments were conducted after studying the in silico or computational tools for that particular atorvastatin, and then that molecule came into existence. Just a minute. Now, this is what we can do. So, being a responsible pharmacist, again, we have got a tremendous space, we have got a tremendous opportunity to work upon the drug like molecules, the data of which is at your hand, at the tip of your finger. Simply, there is a need to click on your mouse, and there the information about the drug molecule is there for you. Okay, so the example is what your PubChem, which can be which is a repository, online repository of several drug like molecules, and simply you just select a drug like molecule, make a change. Now again, it is not necessary to do a experiment in the laboratory. By the knowledge of the structural activity relation, again There are several online tools, several online software are available, and these softwares can make a change in the structural conformation or structure of that particular molecule, and you can have drug like another molecule which is resembling with the standard. Okay. So only you need to have a knowledge of the chemistry and knowledge of the orientation of the chemical structure in the space, and based upon that you can have area to be explored. One more database is available, that is the chemical structure database, the CSD, which contains a structural database of large number of the organic compounds along with their physiological data in a very systematic manner. There are several examples we can put upon, and here is what your zinc database, drug bank, again PubChem, ChemSpider, Toxinet. Okay, so these are the several, what we can say, the online tools or online repositories which are available online for your um, chemical structure database. Okay, so these database bases are also there for you while selecting a drug like molecule, drug like compound for the selected disease. Okay. One more term which is frequently coming across whenever we are discussing any, say, the in silico tool or the computational methodology for the drug development, and that is what the molecular descriptor. So what is this particular molecular descriptor? It is simply a mathematical representation. Okay, don't get confused that a mathematical representation, that means some huge must be there, but it's not like that only. So this is a molecular property, and it is generated using various algorithms. Okay, so for example, say the solubility, or say the partition coefficient, this can be determined by using the several ways, several techniques, and um, after using the several techniques, uh, the figure which is coming repeatedly, that could be considered. Okay, so the numerical value of the molecular descriptors, that is abbreviated as the MDs, are used to quantitatively describe the physical and chemical information of the molecule. For example, log p, that is what your partition coefficient, which is a quantitative representation of the lipophilicity of the molecule. Okay, so this could be the molecular descriptor. Now, being student, what you can do, where you can explore your studies again, simply you can explore your research in determination of the molecular descriptors. So in the repository, suppose from the PubChem, you have selected one compound, and you can have your the title of your research, that description of the molecular descriptors for building of the molecular descriptors for this compound, for that compound. The example what we have discussed here is the partition coefficient, and similarly, other descriptors should be your say the what your solubility. Okay, so permeability. These could be again the possible molecular descriptors what you are doing usually in your regular practicals, and the same procedures can be used for the determination of the molecular descriptors. Now, what is the use of this molecular descriptors? As we have discussed that this particular repository, that is the PubChem, is available where 70,000 billion compounds are there, chemical, which are having the capacity to show the drug like efficiency. But these are not approved drugs, suppose. But you can select that particular drug like molecule, and you can determine all the molecular descriptors, properties associated with the molecular descriptors, and the same properties or the whatever be the values of the molecular descriptors you are getting, The same values you can use further in various in silico tools. Okay, so how to utilize? We are going to see one practical application for the same in subsequent slide. Now, use of this molecular descriptors. So these molecular descriptors can be useful in performing similarity searches in the molecular libraries, as they can find molecules with similar physical or chemical properties based on their similarity in the descriptors value. As I said earlier, suppose you have got the log p value, or say the partition coefficient of any particular compound approximately about say 1. Okay, so unit value. That means compounds or the standard drugs which are having the log p value up to the unit 1 may have most of the same physicochemical properties. Okay, so the concept is very simple. The use or the uh, application of these molecular descriptors, when you are determining the molecular descriptors, simply you are comparing the values of the molecular descriptors with that of the standard drug. So this is what you are doing, the similarity. You are finding the similarity with the unknown. Okay. So unknown you have selected from the repository, which is a drug-like compound, not drug. It is a drug-like compound, but you want to prove it now as a drug. You want to prove its therapeutic significance, and for that sake, you are trying to make it similar with that of the standard one, which is the established drug, which is the approved drug. And that is the use of this molecular descriptor rather. So you are just trying to make it similar with the standard one. Also, these molecular descriptors can be used in various admit prediction. Admit means what? Your ADME properties along with the toxicology. Okay, so that is the toxicity, absorption, distribution, metabolism, excretion, along with the toxicology. So entire toxicology can also be even determined using these molecular descriptors. This ADME profile simply can be predicted. See, if you are determined, say, the permeability, solubility, or say the partition coefficient, which are very essential properties for the biopharmaceutical, which are essential biopharmaceutical properties, and these biopharmaceutical properties directly get in correlation with that of the ADME as well as the toxicity. So you can directly have a prediction of the kinetics of the drug within the body. So, using these molecular descriptors, you can have a better prediction of the kinetics within the body. Now, for example, this is one more online tool, as I said earlier. So, if you are having some knowledge of the chemistry, then you can go for. I will leave this slideshow because I want to show you demonstration regarding this particular first web tool. Is my screen visible? Is my screen visible?
Yes, sir.
Now, this is the first online tool uh, which can give you some idea about the kinetics of your drug within the body. And simply, the principle of working of this particular online web tool is here you can draw the chemical structure of a particular compound or what we can say the similar compound, okay, with that of the your reference drug, or where you are going to explore your research. So there are different tools are available. So this is even where you can say, just a minute. Okay, so so here these are the different keys are available, and using this key you can build up a chemical structure in this particular box. Now, you see all the buttons are available. So if you select this particular, so this could be the ring, or say this is a benzene ring. Ring can be pasted in this particular window. I'm not going to deliberate this all building of the chemical structure, because this is not my own area. And what I'm going to show you about various files can be chosen, and these files are the particular files. The library of these files is again available online. Ganla madam,
Hello,
hello,
sir. हाँ, sir.
just tell me what appears on your screen.
Use of MDS.
Okay, actually, I'm not showing that thing. I'm trying to get the display. Okay, Kanla madam, what is there now?
Something Swiss ADME.
Okay, so okay, now it's there. See what I did
Hmm.
when
Say,
we were
okay.
discussing about the use of the molecular descriptors. Here is the first example what we are going to take into consideration, that is the Swiss ADME. What is the use of this? Is simply you just copy this link and paste upon the address bar of your web page. So here it is what. So this is a Swiss ADME. What is the use of this particular slide? This is the ever best slide, and being a student, this is a free source, open source, and you can take into consideration for your entire research. Simply you can study one drug like molecule, and this particular how to go. So this is a online repositories available already for the sake of the simplicity, and to just make a time efficient presentation. Already I have downloaded few of the files which are compatible with this software. You just have a look. So this is already I have downloaded from the web again, and this is what the structure. Okay, so SDF file, you can say it as, and the online repository for SDF file is there. You can download the SDF structure for any drug like molecule, or else you can build the structure using several online tools which are there on the available, which are there on the web, and which are available openly. Openly means freely. Now this is a structure, and this structure you can turn it into the smiley form. So this is a molecular arrangement. Simply this is a in a special arrangement in a space, and this is a molecular arrangement in this particular space. Now you just hit on the run button. You will have a complete protocol where all the molecular descriptors, all physicochemical properties, its solubility, all the kinetics, okay, related with what we can say here, the ADME, complete ADME profile, everything can be obtained using this free online tool. And this tool can efficiently give you the information about the structure upon which you are working. Okay, so this is what about, see, even you are getting, what could be the possible size, what could be the possible lipophilicity of the drug, what could be the possible flexibility, what is the orientation in the space, what is the conformational dimensions for this particular drug molecule what we have selected for your research. Okay, but even that complete physicochemical profile can be seen over here. uh, and this is data which can be obtained using this free open source software which is available for everyone or every student. So this is what complete data related with the partition coefficient of the drug under consideration. This is the solubility, all the pharmacokinetics you could see over here. You are getting the information about the how the drug is going to get absorbed. Okay, so what are the different inhibitors, how different, say, the enzymes or the inhibitors they could play, or they could hinder the rate of absorption for this particular drug molecule. Okay, so this was all. Even the log kp, that is the skin permeability, the data for the skin permeability can even be obtained using this particular online tool. Okay, so drug likeliness. So what could be the Lipinski state for that? So whether it has got capacity to act just like a drug. So everything can be obtained from this particular online tool. So this was the first example what we can take into consideration while uh, taking your research into the consideration. Okay. So what are the different online repositories are available, and you can get more data. So what are one of the example what we have discussed. So it was for the determination of the ADME. So simply by sitting on the table, we can interpret what could be the possible absorption, what could be the possible solubility, what could be the possible permeability of the drug, partition coefficient of the drug. Everything we are simply getting by sitting on the table, by taking the help of your web tools. And um, once we are doing this particular research, the same research can be validated by performing a lab experiment. Okay, so it doesn't mean that you have used this online tool in silico tool, you have received all the values and done. You need to validate it. And in order to validate all the data what we have received using utilizing these online tools, you need to conduct few of the experiment. And see, these experiments are not at all uh, conducting like it's not a rocket science. Again, these all experiments are very very simple. See, the solubility, permeability, we are doing in a regular fashion, in a regular way, while conducting our day-to-day practicals or the routine work for our projects. So simply, whatever you are doing, you can take help of these online tools so that more resolution, more clarity can be come across in your work. So one more MD descriptor, one more molecular descriptors web tool is available, which is again widely used. One more thing I would like to clear over here, that most of the time while handling these open resources or say the commercial resources, uh, most of the tools may require the knowledge of the programming, uh, but this is only for few of the exceptions, not for all the web tools which are available, which are open source. everywhere it is not needed that you must have a knowledge of the programming for that particular software. Few of the softwares are very friendly used, and there is no need to have a knowledge of the programming. So simply you can download it. The one of the examples what we have right now, the demonstration is a Swiss Adme. So that is the simplest tool where you can have, there is no need to have any knowledge for the programming. Simply you just copy the structure, you simply download the structure from the online repository and paste it into the given box of the web tool. Automatically you are getting entire data for your drug molecule. Okay, so these are other examples. Okay, so I will paste the this particular slideshow in the description for the YouTube channel, and all these links, all the tools will be there in the description for your uh, digital SOP. Okay, so this is one more interesting example, and here also what you need to do, this is for the chemistry. Okay, so you need to study some if you need to study the chemistry of any particular drug. So this is the SDF file for the theophylline, and if I upload this particular theophylline, I'm getting the orientation of this particular theophylline. Now, why three-dimensional structure? Most of the time, you might have heard that the three-dimensional structure, three-dimensional structure. Why it is so? It is essential because the whatever be the conformation, whatever be the arrangement of the molecules in the space of a particular arrangement of the atoms in a molecule. This is a very precise one, specific one. Even the bond angles are very precise one. The conformation of each of the region of the molecule is again very essential to show the therapeutic activity of the drug. So here the conformational or the spatial arrangement of every drug molecule is very essential to study. Now, what the additional information along with the space structure of this theophylline you could have in this particular another web tool. So here you can have this particular uh, different information or information regarding this particular distributor. So you could get more information about this particular theophylline structure, and this is what all. So this is these are the different properties about which you can get. So I'm not going to, because this could be the beyond the scope of this present discussion. You just get to know, you just try to become a familiar with such kind of the online tools which have been there available freely as an open source. So these all properties can be studied uh, from this particular another freeware which is available online. So just hit on the calculate, you will see all the data, all these properties. Approximately, you won't believe, approximately 1800 drug different properties can be fetched by using this particular material. So most of the properties can be required in our drug research. So okay, so these all are properties. There is facility to download it. So you can download it as a CSV file or the Excel sheet, or even the text file can be downloaded from this particular online freeware which is available on net. Okay, so another molecule, another free tools which are there, just like open source molecular modeling, GitHub. Okay, so see, it's very difficult to let you know about the each. See, I have pasted over here 33 different web tools which are there available online, and you can take the help of these all web tools in you somewhere in your. If you split our research, we can do a basic research in a structure. uh, Although you are a student of the sutics or say the cognosy or say the chemistry or say the pharmacology, you can take the help of the various softwares which can build up a chemical structure. Okay. And this, how to select this chemical structure, again we usually feel that it is very difficult, it's very cumbersome task to select one sort of the particular chemical structure, but it's not at all. So, as I said earlier, you just select the disease, you select the standard therapy, you select the standard drug for the same disease, and according to that particular drug, you search the online repository just like a PubChem or any general database available on the online, and from there you can select a drug molecule which is similar to that particular standard drug, and that's what all done. Rest of the things can be performed by all the online tools. If you are going to select that particular compound, you just make a conformational changes. There is no need to go into the laboratory, and no need to conduct the several synthetic type of the experiment. Again, sit on the laptop, connect with the net, and use the different in silico tools, or use different what we can say, the online freewares. With these freewares will do the job of the chemistry for you. They will orient the selected drug or selected compound so that it must have a more and more similarity with the actual compound. And that's what the role of this particular software. Once you have developed that particular chemical structure, now what you need to do, you just use another freewares or the online tools or the web tools, and these web tools will study its all physicochemical properties, can study all the pharmacokinetic properties, even most of the time pharmacodynamic properties can also can be better assessed using such kind of the online tools. So this is what the beauty of what we can say the online web tools which are available and can be efficiently used in your research. Okay. Now, being student, what I would like to share with you that most of the time we feel that how to what is research. So usually most of the students tell us that something new, something new. So something new means what you can uh, conduct your research in such a way that how to explore e-dragon, how to explore the open well, how to explore the pardel, how to explore this chemdes, okay, how to explore this chemexon. So like that, all these whatever be the online repositories, online tools are available. Even you can go and you can make a review to so that the other students could have the information about the efficient utilization of various online repositories, online databases, online web tools, online in silico tools, which can be efficiently used in the process of the drug development as well as in a drug discovery. Okay, so this was all from my side. If you are having any doubt or query, you can raise. So these were all the references. Even the slide for these references has got much of the information regarding the online tools. Okay. So thank you very much. This was all from my side. If you are having any doubts or query, the session is open for the question and answer session. Otherwise, we'll stop here.
Yes, if anyone is having any questions, the session is open for it. Please, you can.
Okay, we'll stop here.
Sir? Okay, sir.
Okay, sir.
Okay.
Just I request all the participants to open your camera once again. We'll have a photo session and there will be validatory function. Okay, thank you. Check, madam, you can continue.
Sir, I'll continue with the out of time, sir. Yes.
Okay, madam.
Thank you, sir, for this wonderful session. So, on behalf of Satara College of Pharmacy, Satara, I take this opportunity to propose vote of thanks I would like to thank today's eminent speaker, Mr. Karinde, sir. Thank you, sir, for this wonderful session. So, with permission of all the dignitaries, I conclude the session is over. Now, I request Ms. Sheikh Madam to start validatory program. Yes, ma'am.
Okay, sir. Good afternoon, everyone. Good afternoon, everyone. I welcome you all for the valedictory program. So, I take this opportunity to propose the vote of thanks of this valedictory function. At the outset, I am very much thankful to the organizing committee, convener, Honorable Principal Dr. N.H. Alurkar, sir, Chief Coordinator, Mr. K.M. Karande, sir, as well as Coordinator, Mr. M.B. Shinde, sir, Assistant Professor, Satara College of Pharmacy, Satara, for organizing such an amazing seminar. I'm grateful to all the distinguished speakers of today's eSeminar, Mr. Kokate, sir, Mr. Lipare, sir, and Mr. Karande, sir, who has accepted our invitation and delivered very useful and informative talk regarding the computer system applications in pharma industry. I am equally thankful to N.G. Shinde, sir, Assistant Professor, Satara College of Pharmacy, Satara, for his technical assistance. I would also like to thank all my colleagues, Ms. Patil ma'am and Ms. Swati Kandla ma'am, for chairing the respective sessions. I would also like to thank Ms. Wag ma'am for the registration of the participants. Last but not the least, I'm thankful to all the participants who have actively participated in today's session. Thank you so much. With this, I conclude that this e-seminar is over.